as just mentioned, my name is Paul Hayes. I'm from the University of Portsmouth, and I'm not going to talk about pollution, and I'm not going to talk about ecosystem services. So I'm going to talk about cyanophage and their potential for controlling the development of cyanobacterial blooms in freshwaters. I heard from a colleague this morning that any talk that includes a word like potential is almost certainly going to be completely contentless, right? And this may well be the case, but bear with me. So a lot of words to tell you nothing much in particular about this topic.
5 minutes before the end,
Oh, it's all right. I will bring us back on time if I can. So the mass growth of cyanobacteria, such as here in the Baltic Sea, where you can see these nodularia and aphanizomenon growing on the surface of the waters of the Baltic Sea. So these mass blooms of cyanobacteria in brackish and fresh waters cause problems for both the domestic, recreational, industrial and agricultural uses of water, and I don't think any talk is complete without a picture of a dead cow that's drunk too much cyanobacteria containing water, so I thought I would put one in for you. Could have been a dead dog, but I know some people are really quite emotional about dogs, so I thought I'd choose a cow instead. So, real problems then for the management of water bodies, and of course part of the MicroAqua project has been about developing probes and systems for detecting cyanobacteria in water before they reach these high concentrations. Now, the growth of these cyanobacterial populations is initiated and initially controlled by bottom-up factors, by which I mean such things as the nutrient content of the water, the availability of light, the temperature, the mixing regime. All of these things control whether these organisms will grow and develop in the water column, and Tony Woolsby, who's talking after me, will talk a lot more about that, so I'm not going to say anything about that, other than it is possible to manipulate some of those factors to try and reduce the growth of cyanobacteria. So you can strip phosphate out of water, you can increase the mixing depth to reduce the light that's available, and so on. So it is, in theory, possible to control growth in that way, but it can be very expensive and the outcomes are uncertain. So the nature of our work has been to see if we can use top-down, i.e. biological factors, to remove cyanobacteria from the water once the population has developed. So grazers, for example, will remove some cyanobacteria, but our focus has been on viruses as a possibility to use these agents as a biological control mechanism to remove cyanobacteria from the water. Okay. So, in common with all other bacteria and all other organisms, cyanobacteria are infected by a range of viruses, or bacteriophage or cyanophage. All those things mean the same thing in this context. So they're susceptible to infection by these agents, and the most common types are shown here. Myoviruses, which attach to the cells and then inject their DNA through this base plate, syphoviruses that have very long and flexible tails, again with a base plate for attachment, and podoviruses, so much shorter tails that attach to the cells. So these are the three common bacteriophage morphologies that infect cyanobacteria, but there are many others, such as filamentous cyanophage. And once you have viruses of this sort available to you in the lab, then it's very easy to kill a bacterial culture. So here's a not very healthy culture, admittedly, of Microcystis, and this is after infection with the cyanophage, and you can see that the suspension has been cleared, so it's possible in a flask at least to control populations of cyanobacteria. But the question, of course, is, okay, this is easy in the lab. What can we do in nature? Can we do the same in nature and use these viruses to control natural populations of cyanobacteria? So, the steps then in testing this process. First of all, one has to isolate oneself some cyanophage, which is the easiest part of the whole process. So you take your host species, something like Microcystis, you mix it with the water sample, incubate it overnight, put it into some low melt agarose, and then plate it onto an agar plate. And if you have viruses that are capable of infecting the cyanobacterial host, then you get these areas of clearance on a lawn of cells, and these are called plaques, and each of these plaques is a viral clone that you have isolated from the water column. And this is what they look like in real life, just to show that we can actually do it. So this is a lawn of Microcystis, and here are plaques that have developed on the lawn after exposure to these water samples. Of course, not all cyanobacteria can be grown on plates in this way, so sometimes we have to isolate the viruses in liquid cultures. And again, you can see here, this is a control, and then this is a dilution series from the water column. And here in the most concentrated samples, you're definitely getting clearance of the host as it's being lysed by viruses. So it's easy then to isolate the phage. What is more difficult is maintaining their viability in the laboratory, and many of the phage that we isolate we're not able to keep them, but there are a few resilient ones that seem to be resistant to storage in the fridge in the laboratory. So there are issues with maintaining viability, but nonetheless it's possible to generate fairly large numbers of clonal viral isolates. So then what are the challenges of using these viral isolates to control the growth of organisms in the wild, as it were? So what are the challenges then going forward? Well, the first point that is important to note is that populations of cyanobacteria are not clonal. Okay? So even if you have a bloom of a cyanobacteria that is monospecific, so it's all Nodularia spumigena, or all Aphanizomenum flossacui, or Microcystis aeruginosa, even though it's monospecific, there's a lot of genetic variation within the population. So there are huge numbers of genetic variants within a population of cyanobacteria in the wild. And this has been shown for all of the genera listed here. Oh my God, I'll get used to this in a minute. So from Anavina, Aphanizomenum, Microcystis, Planktothrix, so all of the toxic bloom-forming cyanobacteria, they all have this genetic variation that has been characterized by looking at polymorphisms of numerous gene loci within individual filaments or individual isolates of these organisms, and the genes that have been used are just listed along the bottom here. So there's enormous genetic variation within the populations, and that the genetic makeup of the population varies in both space and with time. So it's not a constant makeup within the water column, it's changing. And what we have here, just to illustrate that, there's some data from the Baltic Sea, but it could be from a lake or any other freshwater body. So down here we have a range of different genotypes. It doesn't matter what they are, but these are just a combination of alleles in a particular, to make up a particular genotype. And these are 4 locations within the Baltic Sea. And this is just the proportion of the populations that is made up of each of these genotypes. So this population is 54% genotype 12, 25% genotype 11, and so on. And really the only message that I want you to take home from this is that the proportional composition of the population is different at each of the 4 locations. Okay? So at each point within the Baltic Sea, you get a unique combination of genotypes. So that's variation in space. And also, this shows variation in time. So this is now a single location that has been monitored over a 2-year period. So there's time along the x-axis here, and this is the abundance of particular genotypes. And each of the different coloured lines is a different genotype. And you can see that an individual genotype will become more abundant, and then it will crash, and then it builds up again and crashes and builds up. So there's this variation in the genetic composition of the population as a function of time as well as a function of space. Why is that important? Well, it's important because individual cyanophage almost always only infect a subset of the genotypes within a particular cyanobacterial species. So any one of those individual populations within the water column in space or time will comprise a mixture of susceptible and resistant genotypes. And that's illustrated here. So along the top here we have a panel of different viruses that are able to infect nodularia, and they're either myoviruses or syphoviruses. Down here we have a panel of nodularia strains of differing genotypes, and as you can see there are some nodularia genotypes, such as this one, which seems to be susceptible to any virus that you throw at it, but there are others on here. Okay, this one, not a very good example, but we only tested it with 2 and it was resistant to both of them. Okay, so you've got genotypes that are differing in their susceptibility or their resistance to viral attack, and the viruses themselves can either be very broad host range, so there's an example. So this one, for example, this syphovirus over here was able to infect any of the genotypes that we challenged with it. Other viruses are very restrictive in their host range. So this one, this myovirus, only infects these 2 genotypes and so on. So the host, the organisms that we're trying to kill, vary in their susceptibility to viral attack, and the viruses that we can isolate vary in their ability to infect a range of genotypes. So there are these biological factors then which are likely to impact on our ability to control a population that is actually made up of a whole range of genotypes of this sort. Okay, so that's one challenge, is this biological variability in the material that we're using. Another challenge relates to the fact that all of the experiments that we're carrying out are carried out in very controlled conditions in the laboratory. Okay, so in the laboratory we're able to carry out infections and follow the course of viral infection. So this is the sort of a typical infection curve. So this is with Microcystis aeruginosa infected with this podovirus MH142. So at this point here we've mixed the virus into a subset of the cultures, and then the control culture just grows normally as you would expect it to do, but the infected cultures start to develop and then they crash and die. Yeah. So we can use experiments of this sort to sort of work out how long it's going to take to infect the culture and cause its death, but also we can work out such things as how many viruses we need to add to a population to make it all crash at the same time. So what we wanted to do is to achieve this sort of total wipeout of the cyanobacterial culture, which means adding large numbers of virus for each bacterial cell, rather than this, where we're killing some of the cells but many of the others are surviving. So we can look at the sort of the latent period needed for the virus to infect and kill the host, and we can look at the multiplicity of infection that is necessary to kill the cells. And just to make the point that this is something about the variation. So this is a second strain of Microcystis, so a different strain of Microcystis infected with the same virus, and you can see that the infection kinetics here are completely different. So with this particular strain, the virus seems to form a stable relationship with the host. So the ability to kill Microcystis then with this virus is going to be definitely influenced by the genotypes of the Microcystis in the water column. Okay, so there are things that we can find out in culture about the kinetics of the growth. But again, this is all done at a single temperature. If we change the temperature for this particular experiment, then you can see at a higher temperature, so this is the original experiment, at a higher temperature, the phage is completely unable to infect the host. Yeah? And that's a problem if you're working in nature, because although we can control the temperature in the lab and control all of the other growth conditions, we sure as hell can't do that in a lake. So there's a real difficulty here trying to take something from the laboratory, where it works well, to the lake, where it's likely to work less well. So, coming then to nature, so there are a number of questions that one has to answer if you're going to be able to use phage as a way of controlling the growth of cyanobacteria. The first thing is how on earth do you prepare the right phage cocktail to wipe out the population that occurs in the lake? Right? To know which phage to use, you have to know the genotypes of the organisms in the lake. Yeah? Unless you know that, it's unlikely that the phage will have much impact on the population at all. Okay? And it takes a long time to work out the genetic composition of the organisms in the lake. By the time you've worked it out and then prepared the right phage cocktail, it's too damn late, right? You've got a cyanobacterial bloom inflicting all of the damage that you were trying to avoid. One possibility is to use phage that have a very, very broad host range, right? And there are such phage. So in the past, we've isolated a single phage that is able to infect several different genera of cyanobacteria. I can't actually read these things here. So this phage is able to infect Anabina, it's able to infect Planktothrix, and it's able to infect Microcystis. So it's infecting 3 of the major problem genera within cyanobacterial communities. And I think the point of putting it on this tree is just to show you how well separated these 3 genera are in evolutionary time. So they've been separate for a long, long time, and yet there's still this one phage that can infect all 3 of them, but it doesn't grow very well. And although it infects all 3 genera, it doesn't infect all genotypes in all 3 genera, so it still has those same problems. Other problems. Okay, it's easy enough to make enough phage to infect 200 mils of culture in the laboratory at a particular multiplicity of infection. I'm not quite sure how one makes an inoculum big enough for a lake. It would take one hell of a Petri dish to gather enough phage for that sort of inoculum. So, I mean, it's possible using some sort of industrial scaled up process to do it, but immensely difficult and immensely expensive. The other problem is, of course, is how do you get it to the right place within the lake? As I'm sure we're going to hear a bit later, the cyanobacteria are not conveniently sitting on the surface all of the time, waiting for you to spray the virus on them. Right? Many of them, these organisms form subsurface maxima, and you would need to get the phage down into the population in some way. So there are problems with the pure mechanics of getting these phage at the right place in the water column, and we don't have a solution to that. So, coming back to the title, cyanophage and their potential for controlling the development of cyanobacterial blooms, I think that the potential is extremely limited. It's a very good idea on paper. I think right at the start of this project, I said it's never going to work. I think we have pretty much proved it's never going to work in terms of big water bodies, because we have tried it in containers bigger than 200 ml conical flasks, and it's difficult to do it. That said, there may be potential for using them in more limited situations. So perhaps in small oriental ponds it might be possible if one knows about genotypes and so on. And it may be possible to perhaps immobilize cyanobacteria in some way, so where you've got flow through, not cyanobacteria, cyanophage in some way, so that where you have flow through systems, you can flow the water across the phage and perhaps kill the cyanobacteria in that way. But I have to say that I've not been convinced. What I must say is that it's been a lot of fun doing this. And I'd like to thank my co-authors who have made it a lot of fun. So that's Siobhan Watkins, who's now in Chicago, working on other types of phage in sewage treatment systems, and Joy Watts, my colleague from Portsmouth. And I need to thank Annette Richer from the University of Bristol, who taught Siobhan how to grow cyanobacteria, and other colleagues in Portsmouth who helped us with aspects that we've not covered today. So thank you very much.
Thank you very much for your very interesting presentation with this innovative approach. There are questions?
Yeah,
Please.
yeah, Paul, I would like to be the Davis advocate. Yeah, sorry.
Grazie.
Normally,
Forse lo deve accendere? I don't know. There is
must be
Use this one earlier.
you can speak from here.
No, normally the Davis advocate is trying to tell you that you cannot do what you would like to do. I will try to suggest the other round,
Okay.
maybe to retrieve. So let's say you have a different proportion of different genotypes in one population. Now, of course, the idea of wiping out completely all the cyanobacteria is impossible. But I imagine that if you infect a mixed population with just one cyanophage that is selective for one particular genotype, I imagine that after a while the other will take over, and so that one will disappear. Now, if you target those cyanobacteria which are dangerous because they're producing toxin or whatever, maybe you can upset the population of the cyanobacteria within a certain water body. As to the size, I don't know if you tried, I'm sure you thought about it, of using the property of the phages, that is to go from one, I don't know what is the explosion volume of your phages, but probably by a series of tanks in which you infect cyanobacteria and you go to the next, at one point you release a very large number of infected cyanobacteria and hope that the nature does it all. Okay.
Yeah. In relation to the first point, so all of the genotypes that I showed within the nodularia community, all of the genotypes in the Planktothrix and Anabina communities, they are all toxic, right? So if you can knock out one toxic genotype, but then another toxic genotype just comes back in, so you've just changed the composition of the population without affecting its impact on water quality. Yeah? And don't forget, the cyanobacteria and the cyanophage have coexisted for God knows how many hundreds of millions of years, and not one of them has won this competition yet. Yeah? And so there's this constant co-evolution. So, like most biological controls, there's a real problem. Yeah, Tony.
Well, I was just going to say
question?
that there are millions of them that have won the competition.
They're the ones that survive. Yeah.
They no longer exist.
Yeah.
More questions? I am not a specialist, but the European Commission underlined the importance to maintain biodiversity to guarantire, to preserve the function of ecosystems, because ecosystem services are furnished by natural ecosystem in relation also to the conservation of biodiversity. To apply this, if I well understand your innovative biosensor approach, there is relationships between biodiversity and the use of this approach in natural ecosystems? I don't know.
Well, the first thing to say is in the natural condition, the viral cyanobacterial interaction actually drives, is one of the drivers of succession and diversity within the community. Yeah, so it's a driver of that. What we're looking to do here is to perturb that in some way and to wipe out the cyanobacteria. So yes, it's going to impact negatively on diversity.
Thank you.